In this video, we will discuss about biofunctionalization of the hydrogel. So, in this paper, we are discussing their hydrogel method that can replicate the in vivo observations in the in vitro studies. So first, they need they did their study to check whether the hydrogel is properly biofunctionalized or not. In order to check that, the first thing that they did is they stain the cells. They stain the hydrogel, not the cells. They stain the hydrogels and check its Young modulus or the stiffness in the dry conditions and also in the solon condition. For dry conditions, they left the hydrogels at room temperature overnight, and they did their and they checked the thickness using the confocal microscopy. So what they have found out in the case of the dry hydrogel, the thickness is around 3 to 5 micrometer for different stiffness of the hydrogel, and for the solon hydrogel, the thickness is higher, around 75 to 80 micrometer for different stiffness of hydrogel, that is for 2 kilopascal, 12 kilopascal, 50 kilopascals. And they also check the effect of this biotinylated CD3 on the stiffness of the hydrogels. So what they have did is they check the Young modulus without biotinylated anti-CD3 and with biotinylated anti-CD3. They do not see any significant difference in they do not see any significant difference in those conditions, in those two different conditions, at the same percentage of or at the same ratio of this acrylamide by acrylamide. This indicates that the biotinylated anti-CD3 does not have too much impact on the Young modulus of the hydrogel. So far, they have, in the figure on the left hand side, we have understood that their mechanical properties, that is the thickness of the hydrogel in dry and solvent conditions, and their stiffness, and the effect of the anti-CD3 or other proteins on the stiffness of the hydrogel. So far we have understood that things on the left hand side. Now we will understand these biofunctionalization properties. So in the hydrogels, in order to biofunctionalize the hydrogels, they use this streptadivin 488, and they did it by incubating the hydrogels with streptadivin at different time intervals from 0.5 to 3 hours, and they have find out that till 3 hours there is an increase in the signals for this streptadivin. After that, it reaches at its saturation stage, and those results are shown in the panel supplementary figures. In this, they have shown the results that streptadivin at different stiffness of the hydrogels lies on like 3 to 4 micrometers above the bottom layer of the hydrogels, and it does not penetrate inside the hydrogels. For different stiffness of the hydrogels, they have checked this thing, and when they need to check this functional activity of the streptadivin, they use this biotin 543 to bind to the streptadivin, and when they check these biotin 543 signals, these signals are also similar to what they have observed for streptadivin, indicates that different density of the streptadivin does not have impact on the functional activity of the streptadivin. So this indicates that the streptadivin is functional when it is bind to the hydrogels, as it can bind to this biotin 543 antibodies that has affinity towards streptadivin. And these things they have quantified in this panel C and panel D also, where we can see that the Z distance is high, or the fluorescence intensity is high, around 15 to 20 micrometer for the Septabin 488, and similar results they have observed for the biotin 543, where the signals are high between 15 to 20 micrometer, indicates that the biotins is bind to the Septabin 488. And when they check this mean fluorescence intensity at different stiffness, they do not see too much difference. So this indicates that for both the cases, that is Streptococcus 488 and biotin 543 in panel E and F respectively, both of them indicates that the stiffness of the hydrogels does not have much impact on this Streptococcus 488 binding efficiency and the functional activity of Streptococcus 488 and biotin 543 binding efficiency to its specificity, that is Streptococcus. So I hope you understood how they have described their physical properties, physical and mechanical properties of the hydrogel and the functionalization of the hydrogels. If you need to understand more about their experiments or their hydrogel characterizations in detail, you can go through the reference that has been provided in the description. By that you can also refer to the supplemental figures. That's all for this video. Thank you for your time.